Hello guys, welcome to another YouTube tutorial on bioinformatics. My name is Vincent Appiah. Today we are going to look at how to perform genome annotation on bacterial genomes. So what is genome annotation? This is the process of identifying, locating, labeling, and assigning functions to genomic features on a genome. These genomic features include protein coding regions, RNA, etc. Genome annotation is usually performed after you've done the genome assembly, and the annotation is usually performed on contigs or scaffolds. Genome assembly generates contigs, and these contigs are further processed to get the scaffolds. So depending on your method or your approach, you could use contigs or scaffolds. There are a number of tools available to perform genome annotation, and they include PGAP, Rust, and Prokka. For today's tutorial, I'm going to demonstrate the genome annotation process using Prokka. Prokka is fairly simple to use. So to use it, you first specify the Prokka command, then you indicate your options, and then you specify your contigs or scaffolds. For this tutorial, these are the options we are going to use. The first option is prefix. This enables you to give a name. I call it the base name. So this is a name that will be given to all the files generated in addition to their respective file extensions. You could also use the outDar option in place of the prefix. So the outDar enables you to specify a directory where Prokka will save all the files generated. The next option is locus tag. This enables you to give an ID which is unique to that particular genome. And it comes in handy if you are annotating multiple isolates. And so it will be easy to look at which genes and which isolates they were predicted for. CPUs enables you to enter the number of CPUs that Prokka should use to perform the annotation. Of course, the higher the number of CPUs, the faster Prokka will annotate your genomes. That is if the information you entered is correct. Kingdom allows you to specify the kingdom of your isolate. And this enables Prokka to do a more refined annotation because it can pick from the database that you specified instead of doing a more generic annotation. For this tutorial, I'm using a bacterial genome, and so the kingdom option, I'll specify bacteria. The next option is add genes. This enables Prokka to add gene features to the annotation. And the last is proteins. Proteins enables you to specify additional genomes. When I say additional genomes, I mean genome that Prokka will use to annotate your isolate or your genome. So let's say, for example, there's a reference genome for your isolates. You could specify that, and Prokka will use information from that reference genome to annotate your isolates. For the dataset I'm going to use, there is a Mycobacterium ostrans isolate. There is a reference genome for Mycobacterium ostrans, which is the AGY99 strain, and so I will specify that as my proteins. Now the format for the proteins is a GenBank, so I'll quickly show you how to download GenBank formats. So if you go to the page of your organism in the NCBI database, let's say I have my reference genome here. I go to send to, I click, make sure complete record is checked. And then I come and check the file, and then there will be a number of formats here. There are a lot of them. We specify GenBank 4. Note there are two GenBanks here, but I'll specify the one with the 4 in parentheses. That's what usually works for me, and I'll recommend that. And finally, you click the create file. So once you click that, download will proceed. Notice that the default name given to what I download is sequence.gb, so you can decide to change it. So I specify a new name for it, and then I download. So that is how you download the GenBank format for your data. Right. So now we can proceed with the tutorial. So I already have my environment set up. So what I will do is to cd to my assembly. So this tutorial is a follow-up from the previous one, which was the de novo assembly. So you can check out that video so that you can better understand this process. Because for microbial genomics, we usually start from the genome assembly. We do annotation in that order. And so this tutorial will also give you a systematic step as to how you can also perform a comparative analysis for microbial genomes. So now we are in the annotation stage. So let me load my packages and environment. Anaconda is a great tool to manage your software, so I recommend it for everyone. Now I have an environment called Bioinformatics where I install all my bioinformatics tools and use them for my analysis. So let's proceed. So let me confirm that I have my files there. I have my GenBank file. I also have my spit out. This is where I'll get the context. So let me confirm context file is there. Great. So we have it here. That's a context.faster. So now we are ready to go. Let me clear the screen first with this command. Now, if you just type Prokka, you have this information showing up, also telling you what options you can choose for your Prokka annotation and how Prokka is used. So you can always use Prokka to do that. Now let's proceed. So to do the annotation, type my Prokka. I specify my prefix. I'm using MA25. I specify my local stack. Should be in lower caps. I also give it the same MA25, but you can change it. I mean, it's all about your preferences. CPUs. My server has over 20 CPUs, so I'll specify 24. Kingdom is bacteria. Gens flag is added. And I specify my proteins, which is AGY99 ABK. And then finally, I indicate my context, which is in the spit out directory. Right, so now we can proceed. So once everything is okay and Prokka accepts, this is what you see. It starts doing the analysis. Now Prokka uses a number of tools and algorithms to perform the annotation. So what this means is that these tools or algorithms individually perform the annotations or predictions, and then Prokka finally merges all these information into single files for you. That is what Prokka does. So depending on your computing power and then the size of genome, Prokka takes a couple of minutes to perform the annotation. And so as I indicated, if you have more CPU cores, you could add that to the information and then it will make the annotation process faster. So that is how Prokka does the annotation. Great. So now the annotation is complete and that is indicated by this section here. Of course, if there were any errors or warnings, Prokka will still display it. And a log file is also generated at the end of the process so that you can manually check them. So now let's confirm. Because we specify prefix, a directory with that name will be created. So let's check. And that is what we have here. Let me make the screen nicer. And that is what is created here. Now let's see the contents of that annotation. So at the end of the annotation, we have all these files being generated. Of course, I'll give the link to the Prokka homepage. You can check the meaning of these files. So each of them stores a particular data format. Some store dataset for gens, some for annotations, log files, and error. Okay, so those are things we need. So I'll just quickly go over with how we interpret the annotation. So this file stores the coding regions, the .ffn stores the gene, GenBank stores the annotation information, and then others. So to interpret your annotation, you need to know what you are looking for, and you need to know which files those informations can be found. So for example, if I want to find the number of protein coding regions, then I can find them here. Of course, there's another file here that gives summary, each of the features, and then it lists them. So you can also have a look at this one quickly. But for this one, I'm going to use this and then this. So you can use some Unix tricks and commands to find some informations of the annotation that we did to help us interpret and understand our genome. So if I want to find the number of protein coding regions, I can use grep and I specify a directory, then the file, which is .faa. Now, because I want to count, I have to use the pipe command and then add the wc -l to count them. And so once we do it, we have this. So we have 5,421 coding regions. We can also find the number of genes that were predicted. So for that one, you can find it from this file, that's the .ffn. So I'll still use the same grep to give me the information. That is 5,475. Okay. Of course, you can list all of them. So to list them, it's still the same grep, but you don't add wc -l. Of course, it will be numerous. So usually, I prefer not to list all of them. But you can just list a few of them here. Okay, I'm going to list just 10 of them. Great, so we have some of these predicted genes here. So you can see that we have all of them here. Notice these IDs. That's the locus tag we gave it. So the locus tag and it indicates these other ones for labels. Also, of course, this tells you that the MA25, this gene was predicted for the MA25 isolate because that is the locus tag we use for it. Right. Now, you can also do some other activities. For example, if I want to find how many hypothetical proteins identified. I could also use the same grep command and then look for it from here. If it's a protein, you can choose to use the FFN or .FAA. So let me just use the FAA. Then I count them. So that's 2,595. We could also look for insertion sequences. So there's one that I want us to check. That's the IS2404. So we can check if there are some of them there. Because I will list them. Yeah, so we have them listed here. So we have a number of insertion sequence also there. If I want to count, I'll just add that. So we have 19 of them. Right. So the annotation you perform helps you to understand your genome and then its genomic makeup. Okay, so it can even help you to differentiate between maybe a wild type strain and maybe another strain that was found in a different community, probably where there is a disease or not. Okay, and also help you to understand the evolutionary story of your genomes. So these are a couple of interesting things you can find out through the genome annotation. We are going to go into details with this one in a later tutorial, but for today, it was a demonstration to show you how to perform the annotation with the Prokka tool. So I'll end my tutorial for today and then I'll see you again next time. Bye bye.